Welcome to the Research Room SA. My name is Shriyans Thipparthi, and I'm joined today by my co-founders Vijay, Vishal, and Vivaan Achar off camera. We are three students from Health Careers High School in San Antonio, and our show is all about sitting down with the brilliant researchers of our city, such as yourself, and having real conversations about the science happening right here in our backyard. Today, we are incredibly honored to be joined by our very first guest, Dr. Manpreet Semwal, an assistant professor of biology at Our Lady of the Lake University here in San Antonio. Dr. Semwal's research on immune aging and how the thymus maintains immune tolerance has been published in some of the most respected journals in the world, including PNAS and Nature Aging. Dr. Semwal, thank you so much for being here today. And to get us started, can you tell us a little bit about yourself and what drew you to biology and research?
Sure. Hi, Shriyans. Thank you for having me here. As you mentioned, my name is Dr. Manpreet Semwal. Currently, I'm an assistant professor at Our Lady of the Lake University. I received my PhD in immunology under the guidance of Dr. Anne Griffith in UT Health Science Center San Antonio. And my work was focused on understanding how ROS and autophagy are important in T cell development and autoimmunity. Then I did my postdoc in clinical research under the guidance of Dr. Nicholas Musi at the Barshop Institute, where I use single cell RNA sequence technique as one of the techniques to understand how immune cells and other cells are affected during the aging senescence and during diabetes, obesities, and even in aging human patients or participants. Yeah, and right now I am, as I said, I'm teaching at Our Lady of the Lake University. I do research I have two students, and we are working on this fascinating forensic genetic studies where we are going to be are seeing the impact of degradation of both mitochondrial and nuclear DNA over time in some animal tissue samples.
We wanted to talk about your work, and it focuses on the thymus, right? Can you walk us through what it does, why it matters for our immune system, and why you chose to focus on this specific problem?
Sure. So thymus is very underrated organ. It is an organ which is present just above our heart. And it is a really important organ for T cell development. So if you talk about what T cells are, they are special kind of adaptive immune cells, which have various functions, but main functions is fighting viral cells. They fight cancer cells. They also help B cells to help them for their function. And also they're really important in autoimmunity. So my work focused on reactive oxygen species, which is usually given a bad name. Everybody thinks because ROS is a stress factor that ROS is bad, but it is not always the case. Again, I like to say that think about ROS as a fire. Is fire bad? So somebody would say, oh, it's bad,
Yeah, we would
but
assume it
if
to
you
be
don't
bad.
have fire, yeah, you cannot cook the food, right? So you need that balance. So through my PhD thesis, basically what my work showed that if you do not have enough reactive oxygen species early on in the mice thymus, if you restrict that, then these T cells in the mice that develop, they can be autoimmune. So that means you need that fire basically to train the T cells so that they can not only effectively kill the virus, but also they do not attack body's own cells. And as I said, thymus is not everybody knows about it, but I think it's a very important organ, which is important for T cell development, and that's what drew me. And my mentor, I was really fascinated. She was really amazing mentor, and the work I thought was really important, and that drew me into focusing on to that.
And then you study a process called autophagy, correct? Can you explain what that is in simple terms to the audience and why it's so important and needed to be studied?
Sure. So think about autophagy as body's cellular recycling system. So again, one analogy I tell my students is if you're thinking with my project everything, think about that every cell in your body is a restaurant, right? So now this restaurant which is cooking, and the T cells go around checking, is the restaurant okay? So usually when the restaurant is making, it shows, okay, we are making pizza, and show the snapshot outside. and the T cells comes and say, oh, hey, everything looks good, and they like keep on moving. So what autophagy in general, autophagy is really important in recycling. Just think about, when you have a lot of built up in the cell. You don't want it because it becomes inflammatory, it can become toxic. So autophagy is a very clean process of taking that waste materials or recycling it so that you can keep using these macromolecules. Now, if you talk about like why it is important in T cells, so again, think about, let's say this restaurant was attacked, like, by somebody who's now forcing the restaurant to make ammunitions, right? So what will happen, that byproduct will be processed by autophagy, again, the cleaning crew, and will be thrown out. So when these T cells, they are coming and they will look at this restaurant and say, we see some ammunition parts. This doesn't look normal. So that gives them a clue that there is something like an invader inside this restaurant. So we call it as antigen presentation. So autophagy in immune cells is really important for presenting the antigens to the T cells, because a T cell cannot identify the cells. They need to look at what's going on inside the cell, and autophagy provides it. So it's a really important process to like present antigens.
And then I believe you touched on this with your like analogy to the fire, but we wrote that like we were writing about and talking about how your PNAS paper and how it showed that thymic cells like deliberately maintain low levels of an enzyme called catalase, correct? And then it keeps hydrogen peroxide, like low levels of catalase keeps hydrogen peroxide elevated. And then most people hear hydrogen peroxide and they immediately think damage, like as you described with the fire. Can you explain why they specifically need that chemical environment to like function properly?
So the way this whole process works is we have figured out a connection that high ROS levels are needed for autophagy function, and like many different papers have like shown this, like in the literature, and then there are some literature that had also shown that autophagy is needed for antigen presentation. Through our mouse models, we had like different mouse models like catalase, transgenic, and higher autophagy mouse level mice and all. And what we have shown is when you have low catalase, when you're born with it, you have this high ROS, and that fire, like ROS, is needed for autophagy. So again, think about like you need fire to cook, and then you have more cleaning crew, and they can present more antigens to the training T cells. And when this cycle keeps repeating, what will happen? Your T cells come to know, okay, so these are my body's own cells. I don't need to attack it. It's kind of like showing them, is this civilian? Is this like invader? And they have to, make sure that they're only cleaning the invaders. And we tried like having a catalase transgenic mice. When you bring these ROS levels down early on in the life, you will not have fire. When you don't have fire, autophagy is not elevated. So the T cells are not shown what civilians look like. So when they do not have the practice, what happens is in these mice, the T cells onset autoimmunity, and the mice that have low levels of ROS early on are more prone to autoimmunity early stages. So we need that high ROS basically to have less autoimmunity, basically to train the T cells better.
And then you referring to like mice models that you mentioned, right? You then showed that like removing the environment caused mice to develop the autoimmune symptoms, right? And then like you had a follow-up experiment that was able to like rescue that result. And then And then can you walk us through what happened and what that meant for your understanding of immune tolerance?
Sure. So I think we used a couple of mice models. I had a catalase transgenic mice model, which was low ROS levels. And when we compared that to wild type mice, cat transgenic mice were more prone to autoimmune diseases, right? So then we tried to think, okay, so if I have catalase transgenic mice, but if I can increase autophagy in these mice, and that's how we introduced Becklin knock-in, we were able to remove that autoimmunity, which shows us that you need high ROS to maintain higher autophagy. But if you still are able to reach that higher autophagy levels, you can still reduce the autoimmunity. So autophagy I don't know if you are aware, it's one of like really important factors, not just in immunity but also aging. So that it has been like well known in literature that by increasing autophagy, and also the Becklin mice I'm talking about, they have like continuous higher autophagy. They live much longer than their, not much longer, but longer than their other wild type mice, which shows us that autophagy is not only important in longevity, but also keep training the T cells. So that's
So,
how we use different mouse models.
like
Yes.
autophagy, more more efficient autophagy is like directly proportional with like better aging, right? That's what.
Exactly. Yes. Healthy aging. Yes. And there are, I think, for example, we talk about intermittent fasting, right? Why
Yes, yeah.
intermittent fasting increases longevity? Because it triggers autophagy.
It
It's
makes
through one of those
okay,
pathways.
okay.
Yes.
And then another one of your works talked about how the thymus like shrinks up, like it the thymus starts shrinking after puberty, right? And then by
Mm-hmm.
the old age it becomes I believe it was 90%,
10%.
10% of
10%.
it
Yeah.
what it originally
By
was,
70.
right?
Yeah.
How does
Yeah.
your work help explain why our immune system gets weaker as we age?
So, as we age, as you already mentioned, the thymus, it's one of those organs that atrophies really fast,
Mm-hmm.
like in the body. And as I said, by the time you reach 70 years of age, 10% of thymus is left. And think about, as I said, thymus as a training school for T cells, where you are constantly training the T cells to do two things: identify the threat and do not attack your civilians, right? So when you have this downsizing of the school, what will happen, even if your body is making more T cells, they are not getting efficiently trained. And it is known that one of the reasons is, again, let's start with the fire. You started the fire, the ROS, to train the T cells, but as you grow, the fire becomes bigger and bigger. What's going to happen?
It's gonna
It's
burn
going to
it
burn the
down.
house, right? So because of that bigger fire, the ROS, the thymus starts atrophying. And so there are a lot of research that has talked about like reversing the thymus, which is not as efficient, but we talk about slowing down the thymus atrophy. And one of the ways you can do it, and we saw this in our catalase transgenic mice, if you keep the ROS levels down as you age, you control the fire, then you are able to slow down that, like, thymus atrophy. So we are also thinking about how in our elderly participants or patients, we can make sure they have, again, you cannot say, oh, I'm going to have 100% thymus throughout the life. It's not possible as of now, but how can we slow down that? And one of the ways you can do is by bringing down the ROS levels after a certain age. So maybe like people have thought about taking vitamin C, or some other ways or antioxidants to keep the ROS levels down over the time. So to improve that thymus training or T cell training in the thymus.
Mm-hmm. And it would allow you to like maintain better thymus function, right? Like as long
Exactly. Yes.
as you
Yes. So
age.
reversal of it's not possible, but slowing down or healthy aging is something that I think we all are like looking into.
And then we wanted to talk about a pivot in your work. Like,
Mm-hmm.
during your postdoc, you shifted into like what seemed to be a completely different area, using a technology called, like, as you mentioned, single cell
Single cell.
RNA sequencing, right? To study how like fat tissue changes as we age. And then what made you take that leap, and how does it connect back to your thymus work?
Sure. So, like as I said, like my PhD work was mainly in mice in immunology, and for my postdoc, I wanted to do also like do the clinical part of it, like actually work with human participants, and I got this wonderful opportunity with Dr. Musi, who was working with obesity, diabetes, aging studies, but also he needed to somebody who can explore the immunology side of it. So single cell RNA sequencing, the way I would explain is, so again, when you are looking at analysis of the whole cell, again, think about 20 people singing in a chorus, right? So like you hear a combined voice, and then you can say, oh, the song is high pitch, low pitch, it sounds good, it doesn't sound good, right?
Mm-hmm.
Now, single cell RNA sequencing is where you look at the gene expression at every single cell. So again, think about this whole 20 people singing, and in single cell RNA-seq, you go and listen to individual person's voice, and then you can make a quick distinction. Oh, this person is amazing. This person is doing like this note better. So with single cell also becomes really important when you're talking about immune cells that are not in majority, and everybody's singing in chorus. So you say, okay, the inflammation is turned on, but you don't know what kind of immune cells have high inflammation or low inflammation. So in our aging studies, for example, when we were doing some drug interventions, it was really important among the immune cells to go and see, okay, so among T cells, we have this Treg population, which are in decreasing the overall inflammatory markers, and they are doing the 90% of the job. That means we need to focus on these cells if we have to, do this intervention. So a single cell becomes a really powerful tool when you want to take this like whole picture and just focus into individual cell and trying to focus what's happening at the gene expression level.
And then, like a paper you co-authored in Nature Aging earlier this year shows that a specific hormone can actually preserve the thymus and keep it functioning in older mice. And then this is what we were talking about, right? Like reducing ROS levels and like prolonging thymus function.
Yeah.
Do you think we are getting close to something that could slow down immune aging in humans themselves?
Yeah, so it's again like when we talk about these at the animal or human or mammalian level, we have to understand there is a lot of things happening. So just tweaking one or the other thing is not enough. Like and if sometimes when you turn on like something really fast, like switch it on to higher expression, you can have other casualties as well. So as well as like hormones like are known in humans, it is known that women are more prone to like autoimmunity, and one of the factors is estrogen. But does that mean you want to deprive women of estrogen so that they will have less autoimmunity? No, right? So again, you have to like find a way how to balance. And again, to answer the second half of your question, how close are we at? I think there is amazing, tremendous progress, but there is not like one straightforward answer. Everybody is working in scientific community to see how can we again, we cannot reverse the aging, we cannot live immortally, but the main idea is how to give healthy aging, or like make sure our elderly populations have better immune system so that they can fight the infections better. So yeah, so I think it's still ongoing process, but we are discovering amazing work like every year. It's amazing how immunology and other fields are growing, especially because of the technologies we have today.
Right, yeah. And then, Dr. Semwal, this has been an amazing, incredible conversation, and honestly, such an amazing way to kick off our show. And before we let you go, we wanted to ask you, what are you most excited about in your research right now, and what do you see yourself researching in the next 5 to 10 years?
Sure. So one thing I'm really, and I should say like one thing that has really excites me is like I always follow my data. I don't like try to reserve my niche that I want to work only with this like mice only or thymus only, because I think you should go, at least I believe, I go where the data takes me and it gives me some of the discoveries or possibilities that I didn't even think of. So as I said, my PhD, like my postdoc work, we are still like analyzing the data and publishing like amazing work. There were like a lot of like other studies also that I did, which is ongoing. And then currently I'm working with two of my students, and we are doing forensics, genetics, immunology kind of a research in which we are trying to what kind of DNA and nucleus as well as mitochondria can not be degraded, or we can tie it to make it impactful in human cold cases, and that's something I'm really excited about. So again, I want to talk about my journey, how I started with mice, but then I'm like moved to humans, and then now I'm moving to forensics. So
yes.
I just want to say that like your skill set is not restricted to one field. You can like take it anywhere. Just go where the data takes you. And if I have to give, I know you didn't ask for this, but if I have to give one advice, since you're high schoolers, and if you ask me if there is any advice I would like to give it to a high schooler, I would just say that do not get disappointed. You have to start your research somewhere. And if I wish somebody had told me you don't have to figure out everything in one day, like, slowly start understanding the techniques, find a good mentor or a place where it teaches you a lot of like techniques. For example, at OLU also, we are thinking to start a program for high school students where we are going to give them some like a 5-day workshop for, like techniques and tools, and that,
so they can apply it to like whatever their respective
so that they
thing,
can,
right?
so they can have that idea of what research looks like, and hopefully to create a spark so that they can go from there. So I think it's really important to start somewhere and go from there.
Wow. Thank you so much, ma'am. Thank you for your
Yeah.
work and then your willingness
Sure, no
to
worries.
be on call with us. Thank you.
Yes, wish you all the best.
Thank